Bismillahirrahmanirrahim. In lecture number 3, we will discuss about some common carbohydrates one by one, and each carbohydrate we will describe. I will describe you about different kinds of carbohydrates that are very much common. Uh, so let's start with our first carbohydrate. So, the first carbohydrate that I am going to talk about is ribose. Ribose is actually is an aldopentose. It means it is a monosaccharide containing 5 carbon atoms. So that is why. So if we talk about its ring has 4 carbon atoms. So the ring is furanose. But overall, the ribose have 5 carbon atoms, and it has an aldehyde functional group at one end. That is why it is called aldopentose. Aldo due to aldehyde group, and pentose means it has 5 carbon atoms, but 4 in ring, 5 in total, and 4 in ring. So that is why it is called an aldopentose. So in open chain form, there is an aldehyde functional group at one end. That is why we call it aldo. It is a simple sugar with molecular formula is C5H10O5. It means it has 5 carbon atoms, 10 hydrogen atoms, and 5 oxygen atoms. Structurally, the ribose, as it is a partial rearrangement of arabinose. So ribose is very much similar to the arabinose, but they differ from each other at the carbon number 2. So that is why we call it the arabinose, of which ribose is an epimer at the 2 carbon. So they are epimer. Epimer means they are differ from each other at carbon number 2. So that is why the ribose and arabinose are very much close to each other structurally, or the molecular formula is also the same for arabinose and ribose. Further, the D-ribose, it is naturally occurring form of ribose is D-ribose. Is a component of the ribonucleotides which build RNA. So the building blocks of RNA are ribonucleotides, and that ribonucleotide are D-ribose, not L-ribose. That is dextrorotatory ribose. So D-ribose alternates with phosphate groups to form the backbone of the RNA polymer and binds to nitrogenous bases. So these D-ribose are actually the building blocks of RNA polymer that makes to form RNA by making bonds with the phosphate groups and the nitrogenous bases. And further, like L-ribose is actually an unnatural sugar. That is not a natural form. That is the unnatural form. That is why we are not I'm very much concerned about this L-ribose. So we targeted the D-ribose, that is the naturally occurring form of ribose. So here side by is the representation of the aldopentose ribose, where you can see the ring contains 4 carbon atoms, and total there are 5 carbon atoms, and at carbon number 2 the hydroxyl group, at carbon number 2 the hydroxyl group have different position if we compare the ribose to the arabinose. After ribose, the second one is the deoxyribose. As the name indicates, deoxy means it has less oxygen atom as compared to ribose. So let's start with the deoxyribose, or 2-deoxyribose. Is a monosaccharide with the formula C5H10O4. So if you can talk about 2-deoxyribose, it means it have one less oxygen atom as compared to ribose at carbon number 2. So that is why we call it 2-deoxyribose. Its name indicates that it is a deoxy sugar, meaning that it is derived from sugar ribose by loss of an oxygen atom. In deoxyribose, the one oxygen atom is less as compared to ribose. As a component of DNA, 2-deoxyribose derivatives have an important role in biology because these are the building blocks of DNA, and DNA, as we all know, is a genetic material that goes on for the continuity of life and genetic information. So it is very much important in heredity as well. So, the absence of two hydroxyl group in the oxyribose is apparently responsible for the increased mechanical flexibility and more stability of DNA as compared to RNA. That is why the nature chose the DNA as the hereditary material, because it has less oxygen, or it has less hydroxyl group as compared to ribose. So having less oxygen, it is less reactant or less reactive as compared to RNA. So having less reactive, so it means it is more stable. So due to this stability, nature chose the DNA for the genetic information or genetic material to that pass on from generations to generations. So that is why, due to less oxygen atom, it is more stable. Further, deoxyribose is generated from ribose 5-phosphate by an enzyme called ribonucleotide reductase. So it is generated from ribose 5-phosphate by the action of this enzyme, that is ribonucleotide reductase. So here on the top right corner, the graphical representation on the structure of the deoxyribose, we can see at carbon number 2, and there is apart from hydroxyl group, there is hydrogen atom. So that is the difference, the main difference between the ribose and the deoxyribose. The third carbohydrate that we will discuss is cellulose. Briefly, Xylose is a ketopentose, meaning it has 5 carbon atoms and a keto group, except aldehyde, there is a keto group. So it is a monosaccharide containing 5 carbon atoms, which including a ketone functional group in its formula. So it has the chemical formula of C5H10O5. In nature, it occurs in both the L and the enantiomers, and it has an also important role in some of the pathogenic bacteria, and essential role in pathogenic bacteria as well. So this xylitol is also a simple carbohydrate that have a ketone group, except aldehyde. After xylulose, the next one is xylose. Xylose is derived from a Greek word xylon, which means wood. So it is a sugar that is first isolated from the wood. So based on this, the xylose is also called wood sugar, because it is derived from, or it is obtained from the wood. It is the major component of lignocellulose, and like cellulose is the second most abundant sugar or the polysaccharide in nature. First one is glucose, the second most abundant sugar in nature is the cellulose. So it is a major part of the lignocellulose as well. It is a monosaccharide of the aldopentose type, meaning it has a 5 carbon atoms and have an aldehyde group, so that is why it is called aldopentose, which means which means 5 carbon atoms and includes an aldehyde as a functional group. So it is an aldopentose. It is derived from hemicellulose, and one of the main constituents of biomass. Like hemicellulose is one of the main constituent of the overall biomass of a given plant. Xylose is the main building block for the hemicellulose xylane, which comprises about 30% of some plants. So this hemicellulose, most of the plants have this hemicellulose makes about 30% of overall biomass of the plants. Stereochemically, xylose are structurally similar to glucose, but the difference is there. Like glucose is of 6 carbon atom, xylose is 5 carbon atom. Otherwise, both are structurally both are same. The next one is D-Glucose. Glucose derived from the Greek word glucose, which means sweet. Glucose is the simple product of photosynthesis, and that is why we also call it the simple sugar, which have molecular formula is 6S12O6. So glucose is mainly made by plants and most algae during photosynthesis, or even the photosynthetic plants, even their lower plants or higher plants, from water and carbon dioxide using energy from sunlight. So it is the most important source of energy for all the organisms. So there is a primary source of energy, we can say. And the after photosynthesis, this is the first product that is accumulated as a source of energy. So glucose circulates, like we consume glucose, it circulates in our blood, and even the consumer's blood. So that is why the glucose is also called the blood sugar. The naturally occurring form of glucose is D-glucose, while L-glucose is produced synthetically in comparatively small amounts, and that is why it is of lesser importance as compared to D-glucose. The description about D and L we have already discussed in our previous lectures. So continuing with glucose, the glucose molecule can exist in an open chain, that is called acyclic form, as well as it can be in a ring form, that is the cyclic form. D-glucose is also known as dextrose, that is occurs vitally in nature, but L-isomer or the L-glucose does not occur in nature, that is the synthetic form of glucose. In animals, glucose arises from the breakdown of glycogen, and the breakdown of glycogen is called glycogenolysis. Like the glycogen is broken down to get the glucose in animals and even in human beings. Glucose can be obtained by hydrolysis of carbohydrates such as milk sugar, like lactose, cane sugar is the sucrose, maltose, cellulose, and glycogen. So when we after the hydrolysis of these complex sugar molecules, we can get the glucose. Here below are the two projections about the structure of the glucose. Like there is the Fisher's projection, that is in the chain form, and then the cyclic or the acyclic form, then that is the closed chain or ring form, that is the Howard's projection about the glucose. The position in Howard projection, the position of the OH at the carbon number 1 determines the alpha and beta configuration of that glucose molecules, and that we have already discussed in our previous lecture as well. And next carbohydrate is D-galactose. D-galactose is one of the three most common monosaccharides. Like the other two are glucose and fructose, and the third most common is the galactose. The galactose is also known as milk sugar. It is a monosaccharide sugar that is about as sweet as glucose, and about 65% as sweet as sucrose. So it means it is almost equally sweet as glucose, but less sweet as compared to sucrose. It is a C4 epimer of glucose, which means it differs at the position of the hydroxy group at carbon number 4, when we compare this D-Galactose with the D-glucose. A galactose linked with the glucose comprise lactose. So the glucose and galactose combine together to form a lactose that is found in sugar. Galactose is commonly found in dairy products and in avocados, sugar beets, and other gums and mucilage, etc. So there are four isomers are cyclic, in which two of them with the pyranose ring and two with the furanose ring. Like in the right side, we can see the open chain of the galactose and the four cyclic forms. The two are pyranoses, that one is the alpha D galactopyranose and beta D galactopyranose, and the below are alpha D galactofuranose and beta D galactofuranose. Means the pyranoses have 5 carbons in ring and furanoses have 4 carbon atoms in the ring. So uh, they differs in their structures. Uh, when we talk about the number of carbon atoms in the that are found in the ring. So if there are 5 carbon atoms in the ring, we call it the pyranose, and if there are 4 carbon atoms in the ring, we call it the furanose. So there are the four cyclic forms of D-galactose. The next one is Salubios. Salubios is actually a disaccharide with the formula of C12, H22 and O12. It is classified as a reducing sugar. It is derived from the condensation of a pair of beta glucose molecule forming a beta 1,4 bond. ठीक है, two beta glucose molecules forming the beta 1,4 linkage, like the one bond between the carbon number 1 of one pyranose ring and the carbon number 4 of the other pyranose ring. So it is forming the beta 1,4 linkage or the glycosidic linkage. So it can be hydrolyzed glucose enzymatically or with the acid. So they can be hydrolyzed with acid or with the help of the enzymes. Salubios cannot be digested by humans and cannot be fermented by yeast. So they are indigestible. Just like cellulose, the humans can also cannot also digest this salubios. Furthermore, it can be obtained by enzymatic or acidic hydrolysis of cellulose or cellulose material. So we can obtain this salubiose from the hydrolysis of uh, those plants which are rich in cellulose or the cellulose rich material like cotton, jute or pepper. This material is mostly composed of cellulose, so their hydrolysis yields, even though the hydrolysis is acidic hydrolysis or the enzymatic hydrolysis, the hydrolysis of this cellulose or the cellulose rich material produces or gives the salubiose, that is also a polysaccharide, and that is quite similar in its properties, similar to cellulose. Next to salubiose is sucrose. Sucrose is a common sugar or the table sugar that we eat in our daily life. It is also a disaccharide, but in this disaccharide, the one molecule is glucose and the other one is the fructose. So it is a mixture of the glucose and fructose. Sucrose is produced naturally in plants and from which table sugar is refined, like in sugarcane, that is a rich source of sucrose, and we obtain most of the sugar from sugarcane. So that is a typical that we can quote as an example of sucrose. It has a molecular formula of C12H22O11. Sucrose is extracted and refined from either sugarcane or sugar beet or sugar mill, and in sugar mill. So in you can see in sugar mills, mostly sugar mills deal with the sugarcane and the sugar beet. So the sugar mills refine extract and then refine the sucrose from the plant material, plant source, that are the sugar cane or the sugar beets. So they extract sucrose from there, they refine it, and they make finally they make the crystalline form of the sugar that we eat in our daily life. So the hydrolysis of sucrose breaks the glycosidic bond between two monosaccharides, and it yields into the glucose and fructose. So it means the one sucrose molecule is composed of glucose and fructose with the glycosidic linkage. So it is also known as the common sugar or the table sugar as well. The next one is maltose. Maltose is also commonly known as maltobiose or the malt sugar. It is also a disaccharide which contains two D-glucose residues, and those D-glucose residues are joined by glycosidic linkage between carbon number 1, the anomeric carbon of one glucose residue, and carbon number 4 of the other one. Because the disaccharide retains a free anomeric carbon, carbon number 1 of the glucose residues on the right side of the figure. If I can show you here that this one here that I encircle right now, this carbon remains free. So that is why the maltose is a reducing sugar, and the glucose residue with the free anomeric carbon is capable of existing in alpha and beta pyranose forms. So this, because this remains free in this formula or in this form, so this the position of H and OH may interchange with each other, so that is why they can be of alpha or it can be of beta pyranose form. So commonly maltose is produced also in the process of germination, like when the seedlings the seed germinates, it produces in the germination process. That is why the fresh seedlings contains lot of sugars, maltose sugar, and in most of the countries, even our country, people used to cook the seedlings of some of the cereals just to consume this maltose sugar. So if we talk about the sweeting intensity, it is less sweet as compared to sucrose, and sucrose is the standard that we always compare any of these carbohydrate or the sugar with that of sucrose. So it is 30 to 50% as sweet as sucrose. So here below you can see the structural formula of maltose. The next one is hemicellulose. A hemicellulose is also known as polyose. It is one of the it is one of a number of heteropolymer, and it means that have different kinds of monosaccharides or a mixture of different monosaccharides. That is heteropolymer, not the same kind. They are different kinds of monosaccharides, and present along with cellulose in almost all of the terrestrial plant cell walls. So hemicellulose are present along with the cellulose, and it further strengthens the cell walls. The glucose monomers, hemicelluloses, can include the 5 carbon sugars, 6 carbon sugars, as well as 6 carbon deoxy sugars. That is why we call it heteropolymer, because it did not contain the same kind of monomers. It have different kinds of sugar monosaccharides, like they are 5 carbon, they are 6 carbon, or even they are 6 carbon deoxy sugars. That is why it is a heteropolymer. So unlike cellulose, the hemicellulose have a random amorphous structure with little strength as well as branched. So it means the strength of hemicellulose is less as compared to cellulose. and like cellulose are more rigid, they are more compact, but the cellulose is more compact as compared to hemicellulose. So they are easily hydrolyzed by dilute acid or bases, as well as hemicellulase enzymes. So these enzymes can easily hydrolyze the hemicellulose or even the diluted acids or base. Uh, they consist of shorter chains like 500 to 3000 sugar units. Mostly hemicellulose are insoluble in water. The common examples of hemicellulose are xylan, glucoranoxylan, arabinoxylan, and mannal. These are some common examples of cellulose. And below there is the structural representation of the hemicellulose that is present in along with the cellulose. The next one is amino sugars. Amino sugar, a sugar molecule in which the non-glycosidic hydroxyl group is replaced by an amine group. It means that hydroxyl group that is not involving in the glycosidic bond. That hydroxyl group, if replaced by an amine group, so then we that sugar molecule will be an amino sugar because now it contains amino group. So it is also refers technically as 2-amino 2-deoxy sugar. There are more than there are more than 60 amino sugars are known, with one of the most abundant is N-acetyl glucosamine, which is the main component of chitin that makes the chitin. They are present as constituents of glucosaminoglycans, glycolipids, and glycoproteins. Hexosamine means it is a hexose sugar with an amino group, that is hexosamine. Hexosamines are amino sugars where the sugar derivative is a hexose. It means a hexose sugar with an amino group is hexosamines. So the examples, some common examples of amino sugars or hexosamines are glucosamine, galactosamine, fructosamine, and manosamine because it contains the glucose sugar, galactose, fructose, and mannose. That is why if these glucose, lactose, fructose, or mannose contains an amino group, and then we referred as glucosamine, galactosamine, fructosamine, and manosamine respectively. Furthermore, if we uh, talk ananose sugar, that is talk about ananose sugar, that is neuraminic acid. Neuraminic acid is an ananose sugar, 9 carbon monosaccharide, and one derivative of and 9 carbon monosaccharide with an amino group, and that is one of the derivatives of sialic acids, and these sialic acids are involved in the neural transmissions in brain. So the amino sugars, simply we can say the amino sugar is a sugar molecule that contains an amino group, and that is these amino sugars are very much important in biological point of view. The next one are glycosides. A glycoside is a molecule in which a sugar is bound to another functional group. That functional group may be alcoholic group or it may be phenolic or any other group with a glycosidic bond. Before we have talked about two sugar molecules have linked together with a glycosidic bond that forms in disaccharide, oligosaccharides or polysaccharides. But if a sugar molecule is bound with another functional group, and that may be alcoholic, that can be phenolic, that can be any other, with a glycosidic bond, then that molecule we call it glycoside. Many plants store chemicals in the form of inactive glycosides, which can be activated by enzyme hydrolysis. And those glycosides which are inactive can be activated can be active those glycosides can be activated by the hydrolysis. Many plant glycosides are used for medication purposes. The glycosides can be linked by an oxygen, that is O-glycoside, by the nitrogen, that is glycosamine, sulfur, that is thioglycoside, or the carbon, that is C glycosides or carbon dioxide glycosidic bond. So these bonds help to in the linkage of the glycosides. So most importantly, a sugar that we also call glycone in glycosides must be bounded to a non-sugar, that is a glycone, for the molecule to qualify as a glycoside. So a sugar must have bond or glycosidic bond with a non-sugar molecule then we call it a glycoside. Otherwise it will not qualify to be called as glycoside. The sugar group may consist of a single sugar group, that is monosaccharide, or several sugar groups, or the oligosaccharides. Sugar group may be one or it may be many, but those groups must have to linked or bound with the non-sugar molecule. The glycosides can be classified by the glycone, the type of glycosidic bond, and a glycone. So they can be classified as what type of sugar molecules is there, that is the glycone, what type of glycosidic bond, and what type of a glycone or the molecule that is a non-sugar, what is the type of a non-sugar molecule. So they can be classified on the basis of these three these uh, these glycosidic bond and a glycone types. The next one is are mucilages. Mucilages are also polysaccharides that constituted by large molecules of sugars and uronic acids linked by glycosidic bonds. So there are many molecules of sugars, and they are thick, gluey or sticky substances that are produced by nearly all plants and some microorganisms as well, like bacteria, etc. Mainly, these mucilages are composed of carbohydrates with highly branched structures, which include L-arabinose, D-Galactose, L-rhamnose, D-Xylose, and galacturonic acid in various proportions. So these uh, sugar molecules are present in mucilages in different proportions, which actually determines the properties of the mucilages. Plant mucilages can be extracted from a variety of plant parts, including rhizomes, roots, and seed endosperms. So there are various sources of plant mucilages that are produced in various plant parts. So mucilage in plants plays a vital role in the storage of water and food, seed germination, and thickening of membranes. For example, the cacti or other succulents or the desert plants, and even the flax seeds, mean algae seeds, are especially rich source of mucilages. It also contains glycoproteins and other substances such as tannins, alkaloids, and steroids. So mucilages are used in foods as a stabilizer. They are also used as flavoring agents. They are also used as fat substitutes and edible coating of to extend the usefulness of the fruits, or to extend the shelf life of the fruits. They also be used to improve the shelf life of fruits. Uh, the next two mucilages is pectin. Pectin is a structural acidic heteropolysaccharide which contain in the primary cell walls of terrestrial plants and are invariably located in the middle lamella. So this pectin is present in the primary cell wall of land plants, and it is also found in the middle lamella and make the structure more hard. Its main component is galactonic acid, that is sugar acid derived from galactose. The galactose sugar, this galactonic acid is derived from a sugar that is galactose. That is why it is known as galactonic acid. It is produced commercially as a white to light brown powder that is mainly extracted from citrus fruit. The citrus is the main source of pectin, and it is also used as gelling agent. Particularly, it is used in jams and jellies. It is also used in dessert filling, medicines, sweets, and as a stabilizer in fruit juices and milk drinks, and as a source of dietary fiber. It is also a source of dietary fiber. Pectin is naturally found in a number of plants like lemon peel, orange peel, and जो इसमें लेमन के छिलके हैं, ऑरेंज के छिलके हैं, एप्पल पोमेस मीन्स एप्पल का जो है जो हार्ड मटेरियल बच जाए उसमें, कैरेट्स में, सनफ्लावर हेड्स में, गावा में, मैंगोज एंड पपाया. So these are all sources of pectin. The next one is lignin. Lignin is a class of complex organic polymers that form key structural materials in support tissues of vascular plants and even some algae. So they actually provide the mechanical support to the plants, and they are rigid. Even the cells of the higher plants or the bark is rigid due to the lignin present in the intercellular spaces or in the cell wall. So this lignin make the provide the strength or the mechanical support or the rigidity to the higher plants. It is a set of non-sugar molecules acting like a glue to hold the fiber together, or they make compact or they give the compactness or rigidity to the plants. Lignin is chemically bound to carbohydrate materials and occurs throughout the cell wall. It is deposited between and within microfibrils during and after the cell wall thickening process. When there is a cell wall thickening process, it deposits even between the microfibrils and within the microfibrils to make it more hard. The lignin fills the spaces in the cell wall between cellulose, hemicellulose, and pectin components, especially in the tracheids and vessels. They fill those spaces, thus make it more hard. And it also provides a mechanical strength to the cell wall. That's why the cell wall is more rigid due to the presence of the lignin in it. So this is the common features of the lignin which makes the plant cell wall hard. So finally, the importance of lignin. If we talk about the importance overall, the lignin is adhesive to wood, like because due to gluey property, uh, it is adhesive to wood, and it binds the fibers like microfibrils together to make it more rigid. That is why we call it. It provides rigidity or stiffness to the wood. Similarly, lignin is one of the most slowly decomposing component of dead vegetation. जो भी dead vegetation है उसमें जो lignin है ये बहुत slowly decompose होता है. So इसका जो decomposition rate है it is very much slow as compared to the other plant debris. And it also contributes a major fraction of the material that becomes humus as it decomposes. कोई भी जो प्लांट बॉडी है, जब प्लांट मटेरियल है, बायोमास है, जब वो डिकम्पोज होता है, देन इट कन्वर्ट्स इनटू द ह्यूमस. और जो लिग्निन है ये भी ह्यूमस ह्यूमस का कुछ फ्रैक्शन में पार्ट है. इट मीन्स लिग्निन शायद ये मेजर पार्ट नहीं है ह्यूमस का, लेकिन ये ह्यूमस में मौजूद होता है, इवन दो इट इज नॉट मच पार्ट ऑफ दैट ह्यूमस, but it's still present even in fractions. So this is the general importance of lignin. Uh, that is why now we can see how how lignin is important for the plants and especially for this during the cell wall formation. Thank you very much for listening this lecture. I hope so you understand quite well. If any student have any question about this topic, it can he or she can ask me in the comment of this Google Classroom. Thank you very much. Good luck.